Hello everyone. I welcome you all for a brief video presentation on the topic ANOVA, which is an abbreviation to the analysis of variance. A very warm welcome. I'm Bharath M, belonging to third semester Computer Science Engineering with Artificial Intelligence Department of Sri Venkateswara College of Engineering, Bangalore. The subject which I'm taking is mathematics for the CSE stream 3 belonging to of third semester, and the topic is ANOVA, which is a abbreviation to the analysis of variance. Now let us look at the introduction to ANOVA. ANOVA, which is analysis of variance, is a statistical method used to analyze the differences among group means in a sample. It tests the hypothesis that the means of two or more groups are equal. ANOVA provides a way to determine whether the differences between the group means are due to actual differences in the population means or simply due to random sampling errors. There are several types of ANOVA, including one-way ANOVA, which compares the means of three or more independent groups, and two-way ANOVA, which examines the interaction effects between two independent variables on a dependent variable. ANOVA produces an F statistic and a p-value. These both we are going to study it in detail in the coming slides, which indicate the significance of the results. It is widely used in various fields including experimental psychology, biology, and economics to compare means and draw conclusion about populations. This is a brief introduction to ANOVA. Now let us look at the next slide. Here we'll be looking at some of the basic concepts of ANOVA. ANOVA is based on the assumption that the samples being compared are independent and normally distributed. The method calculates the F statistic, which is the ratio between group variability to the within group variability. If the F statistic is large enough to reject the null hypothesis, it indicates that there is a significant difference between at least two of the group means. ANOVA can be used for both one-way and two-way comparisons depending on the number of independent variables being considered. Post hoc tests such as Tukey's HSD or Bonferroni correction can be used to determine which specific group means are significantly different from each other. These are the basic concepts of ANOVA. Next, let us look at what one-way ANOVA is. One-way ANOVA is a statistical method used to compare the means of three or more independent groups to determine if there are statistically significant differences between them. It is based on the analysis of variance, which decomposes the total variance in the data into variance between groups and variance within groups. The hypothesis tested in a one-way ANOVA is that the means of all groups are equal. The ANOVA test produces an F statistic and a p-value. A low p-value, which is usually lesser than 0.05, indicates that at least one group mean is significantly different from the others. One-way ANOVA is commonly used in experimental research to compare the effects of different treatments of or interventions on a dependent variable. It is important for determining whether observed differences are likely due to the treatments or simply due to a random chance. Now, let us look at the key components of one-way ANOVA. The first thing, it is the null hypothesis. This assumes that there are no significant differences between the means of the groups being compared. Alternative hypothesis is the second component in one-way ANOVA, which we are going to discuss now. States that there are significant differences between the means of the groups that are being compared. As we saw there about F statistic and p-value, let us look at a brief description on the F statistic. F statistic is a ratio of the between group variability to the within group variability. It is used to determine if the differences between the group means are statistically significant. P-value is the probability of obtaining the observed F statistic, which is a more extreme value if the null hypothesis is found to be true. A small p-value indicates that the observed differences between the group means are unlikely to occur by chance. Next, let us look what is interpretation, a brief description. If the p-value is less than a predetermined significance level, that is 0.05, we reject the null hypothesis and conclude that there are significant differences between the means of the groups. If the p-value is greater than the significance level, we fail to reject the null hypothesis and conclude that there is not enough evidence to suggest significant differences between the means. Now, that was about one-way ANOVA. Let us look at two-way ANOVA. Two-way analysis of variance, or ANOVA, is a statistical test used to analyze the effects of two independent variables on a dependent variable. It is an extension of the one-way ANOVA, which only considers the effect of a single independent variable. Two-way ANOVA is commonly used in experimental research to examine the interaction between two factors and their combined effect on the dependent variable. It allows researchers to determine whether there are significant differences in the means of the dependent variable across different levels of the two independent variables, as well as whether there is an interaction effect between the two independent variables. Interpreting the results of a two-way ANOVA involves analyzing the main effects of each independent variable and the interaction effect. The main effects indicate the individual impact of each independent variable on the dependent variable, while the interaction effect shows whether the effect of one independent variable depends on the levels of the other independent variable. In summary, two-way ANOVA is a powerful statistical tool for analyzing the effects of multiple independent variables on a dependent variable and assessing their interactions. It provides valuable insights into the relationships between variables and helps researchers make informed conclusions based on their experimental data. This was a brief introduction to the two-way ANOVA. Now, let us look on about the assumptions of ANOVA. First one, independence. One of the key assumptions of ANOVA is that the observation within each group or treatment are independent of each other. This means that the value of one observation does not depend on or influence the value of any other observation. Second one is the normality. Another assumption of ANOVA is that the residuals, which is the differences between the observed values and the predicted values, are normally distributed. This assumption is important because ANOVA relies on the normal distribution to make accurate statistical inferences. These are about the assumptions of ANOVA. One more is the homogeneity of variances. Here, the assumption of homogeneity of variances states that the variances of the groups being compared are equal. This means that the spread or dispersion of the data is similar across all groups. Violation of this assumption can lead to inaccurate results and interpretations. These three were the assumptions of ANOVA. Now let us look on interpreting the ANOVA results. First one is F statistic. The F statistic is a measure of the variation between groups compared to the variation within groups. It tests whether there is a significant difference in the means of the group being compared. A higher F statistic indicates a larger difference between the groups. And another one with F statistic which we saw was the p-value. The p-value is the probability of obtaining a result as extreme as the observed result, assuming that the null hypothesis is true. A p-value less than the chosen significance level, which is usually 0.05, indicates that there is sufficient evidence to reject the null hypothesis and conclude that there is a significant difference between the groups that are being compared. Now, let us look what are the advantages of ANOVA. The first one, let us look at 5 points in total. The first point, ANOVA allows for the comparison of more than two groups or conditions, providing a more comprehensive analysis than t-tests. It is a powerful statistical tool for determining whether there are significant differences between the group means. ANOVA can handle both categorical and continuous independent variables. It provides information on the effect size, allowing for a better understanding of the practical significance of the results. ANOVA can be used to analyze data with unequal sample sizes and different variances. These are the 5 advantages of ANOVA. When there are advantages to any certain things, there is a fair chance of having equal number of disadvantages. Now, let us look at the disadvantages, or also known as the limitations of ANOVA. ANOVA assumes that the data follows a normal distribution and that the variances are equal across all groups. This is a big limitation of ANOVA. It is sensitive to outliers, and the presence of extreme value can affect the results. ANOVA does not provide information on the direction or magnitude of the differences between groups. It does not account for interaction between independent variables. As the name suggests, independent variables, there will be no interaction between the two variables. ANOVA is a parametric test, meaning that it relies on certain assumptions about the data. If these assumptions are violated, the results may be invalid. These are the main limitations of ANOVA. Now, let us see where all ANOVA is applied in real life. The example applications of ANOVA. First thing, let us look at just three now for time being. First thing is psychology. ANOVA is commonly used in psychology to analyze the effects of different treatments or interventions on a group of participants. It helps researchers determine if there are significant differences in outcomes between the treatment groups. The second one example is biology. In biology, the analysis of variance can be used to analyze the effects of different variables, such as different doses of a drug or different environmental conditions, on a biological response. It helps researchers determine if there are significant differences in the response variable across the different groups. ANOVA is used in business to analyze the effects of different factors such as price, promotion, and location on sales or customer satisfaction. It helps businesses identify which factors have a significant impact on their outcomes and make data-driven decisions. Now, before we conclude, let us look on the future trends of ANOVA. Future trends in ANOVA may include advancements in statistical methodology, such as more complex models to handle non-normal data and unequal variances. Bayesian ANOVA could become more prevalent for its flexibility and ability to incorporate prior knowledge. With the rise of big data, ANOVA techniques may need to adapt to handle large and complex datasets. Non-parametric methods may also see increased use, particularly in situations where data distribution assumptions cannot be met. Integration with machine learning algorithms could improve predictive modeling. Interactive and visual tools may be developed to aid in ANOVA analysis. Additionally, meta-analysis techniques may be more widely used to combine results from multiple studies. These trends aim to enhance ANOVA's flexibility, scalability, and interpretability to meet evolving statistical analysis needs. These are the future scope, or also called the future trends of ANOVA, which is the analysis of variance. With this, let us conclude with the presentation. Thank you all for being through the video. See you soon. Thank you.